Today, we are going to do something a little different. Usually, we look at the world around us, history, tech, society, the big macro trends. But today, we are looking inside.
Deep inside.
And I don't mean introspection or mindfulness. I mean physically inside.
We are going microscopic. We're going to the molecular level.
Exactly. We're taking a stack of research, specifically Chapter 68 of a pretty dense, heavy-duty physiological text, and we're going to figure out the biological machinery of life.
It's a fascinating journey.
I want to start with a question, though.
Yeah.
A really simple one. You had a sandwich for lunch, right?
I did. Turkey and Swiss on rye.
A classic.
Classic.
Okay. Now, here is the history that we usually ignore. How does that sandwich, how does it become a thought?
Right.
How does that bread and that meat turn into the actual physical energy that allows you to blink your eye or type an email or, for your heart to beat? Because at the end of the day, we are just combustion engines running at body temperature.
That is a very, very apt analogy. We are chemical engines. And the mission for this deep dive is to decode that chemical engineering inside our cells.
The black box.
The black box. We're going to trace the journey of a single molecule of glucose, that's just sugar, from the moment it knocks on the door of your cell until it is completely burned up to keep you alive.
And the sources we have for this are fantastic. It's this incredibly detailed breakdown of what's called carbohydrate metabolism. It covers everything from how sugar gets into the cell in the first place to how we squeeze every last drop of energy out of it.
Every last drop.
And to start, we have to talk about money.
Money. Where are you going with it?
Well, currency, the energy currency of the body. Because you can't just, shove a piece of bread into your bicep and expect it to flex.
Right. No, the cell doesn't accept bread or even glucose directly as payment for work. It has a very specific currency.
Yeah, and that is?
That currency is adenosine triphosphate, or as everyone knows it, ATP.
ATP. I feel like I remember this from high school biology, all the powerhouse of the cell, memes and everything, but let's actually unpack it properly. Why do we call it a currency?
Think of it this way. The carbohydrates and fats you eat, your turkey sandwich, are like gold bars.
Okay.
They have a ton of value. There is a huge amount of energy stored in those chemical bonds. But you can't walk into a convenience store and buy a pack of gum with a gold bar.
Right. So too big. It's not liquid assets. The clerk would just stare at you.
Exactly. The clerk won't take it. In the cellular world, the little proteins that contract your muscles or the pumps that move ions in your brain, they cannot handle the raw energy of a whole sugar molecule. It's too bulky, too much all at once.
You have to exchange it.
You have to go to the bank and exchange that gold for cash. ATP is the cash. It's the universal medium of exchange that every tiny machine in your cell accepts to do its job.
So the mitochondria, which I'm sure we'll get to, is basically the bank teller changing the gold into cash.
That's a perfect way to think about it, precisely. And just like cash, you spend it, you earn it, you spend it again. The average ATP molecule is recycled thousands of times a day. It's a constant flow.
Let's look at the cash itself then. Visually, what are we holding? What is ATP actually made of?
Okay, so it's a chemical compound, a nucleotide. You start with a nitrogenous base called adenine.
Adenine.
You attach that to a 5-carbon sugar called ribose.
Okay.
And together, those two parts make adenosine. You can think of that as sort of the handle of the tool.
All right. They did as
The business end, the blade, if you will, is a tail of 3 phosphate radicals, hence triphosphate.
The adenosine triphosphate. And the magic is in that tail, isn't it? The source material talks about this squiggle.
The squiggle, yes. It's great. In biochemistry notation, we use a wavy line, a little squiggle symbol, to represent a high-energy bond. And those last 2 phosphate bonds in the chain are incredibly unstable.
Why are they unstable? What makes them so special?
Think about magnets. Phosphate groups are negatively charged. What happens when you try to push the negative ends of 2 magnets together?
They repel. They push apart really hard.
Exactly. They desperately want to fly away from each other. But in this molecule, chemical bonding forces are holding them together against their will. They are under an incredible amount of tension.
Like compressed springs.
Perfectly put. They are like compressed springs just waiting to pop.
So when the cell needs to do something, contract a muscle, send a nerve signal, anything, it just snaps one of those phosphates off.
Exactly. It breaks that high-energy bond. The ATP becomes ADP adenosine diphosphate, di for 2, and that snap releases a usable burst of energy.
And here is where the numbers get really fascinating, because the source points out a huge difference between theory and reality.
Yes. This is crucial. If you look at standard textbook chemistry conditions, in a test tube at room temperature breaking, that bond releases about 7,300 calories per mole of energy.
Okay, 7,300. That sounds like a decent amount.
It is. It's not trivial. But here is the secret the physiology text reveals. Inside the actual human body, because of our specific temperature, 37 degrees Celsius, and the specific chemical environment of our cytoplasm, it's not 7,300.
It's more.
It explodes to 12,000 calories per mole.
Wow. So our bodies are actually turbocharging this reaction. We're getting way more bang for our buck.
We are running high-performance environments, so a full 12,000 calories of energy are unleashed every single time you snap a phosphate off. That is the energy that powers life.
And then you're left with ADP, adenosine diphosphate.
Right. The spent shell casing. And if you're really, really desperate for energy, you can actually snap another phosphate off and go all the way down to AMP adenosine monophosphate. But that's rare. Usually the cycle is just ATP to ADP and back again.
So the whole point of eating, of breathing, of literally everything we're going to talk about today is just to turn that ADP back into ATP.
That is the entire game. We are constantly, ceaselessly recharging the battery. If you stopped making ATP, rigor mortis would set in, and cellular death would happen almost instantly.
Okay, so we know what the goal is, make ATP. Now let's go to the very beginning of the assembly line. Section 1, the entry.
Getting the fuel.
We've got this glucose molecule floating around in our blood after that sandwich was digested. It drifts up to a muscle cell. It needs to get inside to be used. But there's a problem, isn't there? A physical one.
A very physical problem. It's a matter of size. If you look at the molecular weight of glucose, it's about 180.
And is that big? In the cellular world?
In the cellular world, yes, it's pretty chunky. The cell membrane is this lipid bilayer, basically a fatty wall. It has little protein pores in it to let things drift in and out. But those pores have a maximum size of about 100 molecular weight.
Oh. So glucose is 180, but the door is only 100. It's like trying to fit a grand piano through a cat flap.
It physically cannot fit. The membrane pores are just too small. If we relied on simple diffusion, the sugar would just bounce off the cell wall, and we'd all starve to death surrounded by fuel.
So how does it get in? There must be another way.
The cell uses a different mechanism, a more elegant one called facilitated diffusion.
Facilitated. So it gets help. How does that work?
Imagine a revolving door. Embedded in the cell membrane are these large carrier proteins that span all the way from the outside to the inside. The glucose molecule floats up, hits the outside of this protein, and binds to a specific receptor site.
Like a key in a lock.
A bit like that. And when it binds, the protein reacts. It instantly and physically changes its shape. It sort of flips or rotates, and in doing so, it pushes the glucose into the inside of the cell.
And does this cost energy? Does it burn ATP to do this?
No, that's the beauty of it. It's passive. It works by diffusion. It's still moving from a high concentration of glucose in the blood to a low concentration inside the cell, but it needs that carrier protein to facilitate the move.
A revolving door.
Without the revolving door, the wall is solid.
And this is where one of the most famous hormones in the entire world comes into play, insulin.
Yes, this is absolutely crucial. In many of the body's tissues, especially muscle and fat cells, that revolving door is naturally very, very stiff. It's almost locked.
So without insulin?
Without insulin, the rate of glucose transport is painfully slow, almost negligible.
But the source says it increases the rate by 10 times or more when insulin is present?
Correct. When your pancreas secretes insulin, which it does because you just ate that sandwich and your blood sugar is rising, the insulin circulates through your blood, hits the cell, and essentially greases the hinges of these carrier proteins. It unlocks the door.
So if you don't have insulin, your cells don't respond to it.
The hallway, which is your blood, is packed with food, but the doors to the rooms are all locked. The cells are literally starving. This is the tragedy of untreated diabetes. The blood sugar is sky high, but the cells are dying for lack of energy because they lack the key to open the door.
That's a powerful way to put it. However, I noticed an exception in the notes that really stood out to me. The liver and the brain.
Ah, good catch. Yes, the brain and the liver are very special cases. Their carrier proteins are a different model.
And they don't need insulin.
Their doors are always open. They don't depend on insulin.
Why is that? Why the special treatment?
Think about it from a survival perspective. The brain is the command center. It consumes a massive amount of glucose. It's extremely metabolically active, even when you're sleeping. You absolutely do not want your brain to shut down just because your insulin levels fluctuate a little bit between meals.
Right, that would be a design flaw.
A catastrophic one. So evolution designed a failsafe. The brain takes whatever glucose it needs whenever it needs it, regardless of your insulin status.
That makes perfect sense.
Uh,
Always prioritize the CPU. Okay, so we've got the glucose inside the cell. The doorman let it in. Now, what's the very first thing that happens? The source calls it the trap.
I love this part. It really is a trap. The moment that glucose molecule crosses the threshold into the cytoplasm, an enzyme jumps on it. Instantly. If you are in the liver, that enzyme is called glucokinase. In most other cells, it's called hexokinase. Same job, different names.
And what do these enzymes do to the glucose?
They slap a phosphate group onto it. It becomes glucose 6-phosphate.
Why do that immediately? Is it just to start the breakdown process?
Yeah. It serves two brilliant purposes. Yes, it does prepare it for breakdown, that's one. But primarily, it's a security measure. Remember that revolving door, the carrier protein?
The one that let the glucose in.
Yeah. It works both ways. If the glucose concentration inside the cell for some reason got higher than the blood, the glucose could just turn around and diffuse right back out.
Right. We don't want to lose our fuel after all that work getting it inside.
Exactly. But here's the trick. The carrier protein doesn't recognize glucose 6-phosphate. It only recognizes and fits regular glucose. By adding that phosphate group, the cell changes the shape of the molecule just enough that it no longer fits the exit.
Oh, that is clever. It's like checking into the Hotel California. You can check out anytime you like, but you can never leave.
That is the perfect analogy. Once it is phosphorylated, it is chemically locked inside the cell. It's committed to being used by that cell.
So it's trapped for good.
It is. The only major exception are liver cells. They are unique.
Why the liver?
Because the liver is the glucose bank for the whole body. It's not just using glucose for itself. So it has a special enzyme called glucose phosphatase that can pick the lock and remove that phosphate.
And that allows it to, what? Release glucose back into the blood?
Precisely. The liver can dispense sugar back into the blood when the rest of the body, especially the brain, needs it during a fast. But for a muscle cell or brain cell, once sugar is in, it stays in.
Okay, so the glucose is trapped. Now we have a choice. Do we burn it right now or do we save it for later? Let's talk about section 2, the savings account.
Right. We don't always need to burn all our energy immediately. If you ate a huge meal and now you're just sitting on the couch watching TV, you don't need to burn all that glucose right now, so we have to store it.
And we store it as glycogen.
As glycogen.
I've heard of glycogen. It's basically animal starch, right?
Right.
A long chain of glucose molecules.
Pretty much. It's a massive polymer. Instead of having thousands of little individual glucose molecules floating around, the cell links them all together into one giant branching chain.
But here's my question on that. Why bother linking them? Why go through the trouble? Why not just keep them as individual glucose molecules in a little baggie inside the cell?
This brings us back to physics again. A really fundamental principle. Osmotic pressure.
Okay, explain that to me in simple terms.
Osmotic pressure depends on the number of particles in a solution, not on their size.
The number, not the size. Got it.
So if you had thousands and thousands of tiny little sugar molecules floating around in the cell fluid, they would attract water like crazy. Water from outside would rush into the cell to try to dilute all that sugar.
And the cell would swell up.
It would swell up and eventually burst. It would physically pop like an overfilled water balloon.
That sounds bad.
It's fatal, utterly fatal. But by linking those thousands of glucose units into one giant glycogen molecule, you cleverly reduce the particle count from thousands down to one.
So the water doesn't rush in, the osmotic pressure stays balanced.
Exactly. It makes the storage osmotically safe. It's like the difference between filling a room with thousands of loose sheets of paper versus filing them all neatly into one dense cabinet. The room is cleaner, safer, and you can fit a lot more in.
How much can we actually store? What's our capacity?
It varies by cell type. The liver is the main vault. It can store up to 5 to 8% of its entire weight as glycogen. That's a huge amount of fuel density.
And muscle cells?
Muscle cells are more like 1 to 3%. They keep a smaller private stash for their own use.
So the liver is the main savings bank for the whole body?
It is. It buffers blood glucose for everyone. Muscles just save for themselves. They're a bit selfish.
Now here's where it gets really interesting for me. When we want to make a withdrawal, let's say I suddenly see a bear and I need to run, or I just realize I'm late for a meeting. How do we get that sugar back out of storage? The source mentions it's not just the reverse of how we stored it.
No, biological systems rarely work in simple reverse. It's a different pathway. To break glycogen down, a process called glycogenolysis, you need a specific dedicated enzyme called phosphorylase.
Phosphorylase. Sounds like a demolition tool.
It is. It chops the glucose units off the glycogen chain one by one. But here's the catch. Under normal resting conditions, phosphorylase is asleep. It's inactive.
Why?
Well, you don't want your cells constantly breaking down your savings account for no reason. You want that money to stay safely in the bank until you actually need it.
So what wakes it up? What's the alarm clock?
There are two main triggers. Trigger number 1, epinephrine. Adrenaline.
The bear.
The bear, exactly. Your sympathetic nervous system fires, your adrenal medulla pumps out epinephrine, and that chemical signal screams at your liver and muscle cells, wake up the phosphorylase, we need fuel and no BUN.
And the second trigger.
The second one is glucagon. This is more of a gentle, low-fuel warning light.
Not a panic button.
Not a panic button. When your blood sugar drops because you haven't eaten in a while, your pancreas releases glucagon. It travels to the liver and says, hey, the tank is getting low. Could you please release some of the reserves for the brain?
And the mechanism described for this is pretty complex, involving something called cyclic AMP.
Yes, CMP. It's what we call a second messenger. The hormone, either epinephrine or glucagon, is the first messenger. It hits the outside of the cell, but it can't actually get in.
So it passes the message along.
It rings the doorbell, and that triggers the creation of CAMP inside the cell. And the CAMP acts like a foreman.
A foreman, I like that.
It kicks off a whole cascade of reactions, like a row of dominoes falling, that amplifies the signal and finally activates the phosphorylase enzyme to start chopping up the glycogen.
So it's an amplification system.
A huge amplification system. One single molecule of adrenaline hitting the cell can trigger the release of millions of molecules of glucose into the bloodstream. It's incredibly powerful.
So we've mobilized the fuel, we've cracked the safe, we've got our glucose ready to burn. Now we finally enter the furnace. Section 3. Glycolysis. This is where we start to break the glucose apart.
Glycolysis literally means splitting sugar. This all happens out in the cytoplasm, the general fluid of the cell, and it's a 10-step chemical dance.
10 steps. It seems like a lot of work just to split one molecule in half.
It is quite complex, but it's very controlled. We take that 6-carbon glucose molecule and we chemically modify it step by step, twisting it and adding phosphates until it becomes unstable enough to split perfectly into 2 3-carbon molecules.
And those are called?
Pyruvic acid. That's the end product of this stage. 2 molecules of pyruvic acid for every 1 molecule of glucose that enters.
Okay, pyruvic acid. But let's look at the energy tally here, because the source points out something that seems pretty surprising. Glycolysis seems kind of wasteful, or at least not very impressive from an energy standpoint.
It is incredibly inefficient if you look at it in isolation.
Yeah.
First, you have to remember that you have to spend money to make money.
An investment.
An investment. We have to use 2 ATP molecules just to get the process started, to phosphorylate the fructose and glucose to make them unstable enough to split.
So we're 2 in the hole to begin with.
Right. Then, as we split the molecule and it goes through the rest of the steps, the reactions release enough energy to generate a total of 4 ATP molecules.
Okay, so we spent 2, we made 4.
Yeah.
Our net profit is just 2 ATP.
That is peanuts. It's almost nothing.
Yeah, considering we said earlier that the goal is to make buckets and buckets of this stuff to power the whole body.
To put it in perspective for you, 1 single molecule of glucose holds a total of 686,000 calories of free energy locked in its bonds.
Okay.
Those 2 ATPs we just made. That represents only about 24,000 calories of captured energy.
So we've unlocked, like, what is that, just over 3% of the total available energy.
Roughly 3%. The other 97% is still locked inside those 2 molecules of pyruvic acid.
So if our body stopped here, if
Yeah.
we only had glycolysis,
We'd die. It would be like chopping down a massive redwood tree, burning a few twigs for a tiny little campfire, and leaving the massive energy-rich trunk just sitting there on the ground, completely untouched.
so where does the trunk go?
It goes into the main power plant, it goes into the mitochondria. This brings us to section 4, the citric acid cycle.
Also known as the Krebs cycle.
The Krebs cycle, yes. The pyruvic acid molecules leave the cytoplasm and are actively transported into this double-walled organelle, the mitochondrion.
And once they're inside,
The first thing that happens is a little transition step. The 3-carbon pyruvic acid is converted into a very, very famous 2-carbon molecule called acetyl coenzyme A, or just acetyl-CoA.
I feel like acetyl-CoA is the VIP pass for the mitochondria. I see it everywhere in diagrams.
Is the central hub of all metabolism. It's not just carbs. When you break down fats, they turn into acetyl-CoA. When you break down some amino acids from proteins, they turn into acetyl-CoA. It's the universal fuel that feeds into the furnace.
The common entry point.
The common entry point. So this acetyl-CoA enters the cycle proper. What happens next?
It combines with another molecule, right?
It combines with a 4-carbon molecule that's already in the cycle called oxaloacetic acid. And when the 2-carbon acetyl-CoA joins with the 4-carbon oxaloacetic acid,
You get a 6-carbon molecule.
you get a 6-carbon molecule called citric acid, and hence the citric acid cycle.
And it's a cycle because at the very end of the loop, we get that oxaloacetic acid back.
Exactly. It's a perpetual loop. Acetyl-CoA comes in, we spin the chemical wheel, we strip off energy and carbon atoms, and at the end, we regenerate the oxaloacetic acid so it's ready to catch the next acetyl-CoA that comes along. It's incredibly elegant chemistry.
Now let's look at the output here, because I'm still looking for the energy. We get some CO2. That's the waste product we breathe out, right?
Yes, the carbon atoms from the original glucose molecule are finally released as the CO2 we exhale. That's where it goes.
And we get 2 more ATP, 1 for each pyruvic acid that went in.
Just 2, that's correct.
Wait, I'm doing the math here. We're still at 2 net from glycolysis, plus 2 from this cycle. We're at a grand total of 4 ATP. This is still terrible efficiency. We are still missing the vast, vast majority of the energy. Where's the massive payout?
You're missing the gold because you're looking for cash. The Krebs cycle isn't really about making ATP directly. That's just a tiny side effect. It's about harvesting something else entirely.
What?
Hydrogen.
Hydrogen, the simplest element.
Yes. During this cycle, a series of enzymes called dehydrogenases are aggressively stripping hydrogen atoms off the molecules as they're being processed.
Why do we want hydrogen?
Because hydrogen atoms contain high-energy electrons. That's where the real power is. Think of the hydrogen atoms as fully charged battery packs. But we can't just let them float around loose in the cell. They're too reactive. We need a special carrier.
Enter the taxi driver.
The taxi driver, yes. A molecule called NAD plus DINI, nicotinamide adenine dinucleotide.
That's a mouthful. Let's just stick to NAD plus cook.
Less. NAD plus is an electron carrier. It's an empty taxi. It picks up the hydrogen atom becoming NADH and ferries it safely to the final stage of the process. The Krebs cycle is basically a harvesting machine that does nothing but fill up these NADH taxis with high-energy hydrogen passengers.
So the entire purpose of the Krebs cycle is just to load up the trucks.
Exactly. It's all just prep work for the main event. And now the fully loaded trucks are heading to the dam. Section 5, oxidative phosphorylation.
This is where the magic happens. The source says, what, 90% of our total ATP comes from this single stage.
This is the payoff. This is the whole reason we breathe oxygen. This is all occurring in the inner membrane of the mitochondria, those folded shelves you see in diagrams, which are called cristae.
And the mechanism is, it's wild. It's called the chemiosmotic mechanism. It sounds more like physics or engineering than biology.
It is sophisticated nanoengineering. The best analogy is a hydroelectric dam. On one side, you have a high water level. On the other side, it's low. If you open a sluice gate with a turbine in it, the water rushes through and the force of that flow generates power.
Okay, I can picture that. A Hoover Dam inside the cell.
The mitochondria builds a dam, but instead of water, it uses protons, which are just hydrogen ions, H+.
Okay, walk us through the steps. We have our NADH taxi arriving at this inner membrane, carrying the hydrogen.
Step 1, ionization. The hydrogen atom is released from the NADH. It immediately splits into its 2 component parts, a proton, H+, and a high-energy electron, E.
They separate.
They separate. Step 2, the electron transport chain. That high-energy electron is immediately passed like a hot potato along a chain of special proteins embedded in the membrane.
These are the cytochromes.
Flavoproteins, iron sulfide proteins, ubiquinone, and finally a series of cytochromes.
Cytochromes B, C, A, and finally A3.
Yes, cytochrome oxidase A3 is the last stop on the line. Now, this chain is crucial. As the electron jumps from one protein to the next, like it's falling down a flight of stairs, it loses a little bit of energy at each step.
And where does that energy go? It can't just disappear?
That energy is used to power proton pumps. 3 of the protein complexes in this chain are actually pumps. And they use the energy from the falling electron to actively shove the protons, the H+ Fes, from the inner matrix of the mitochondria to the outer chamber, the space between the 2 membranes.
So we are pumping protons out.
We are pumping them out against their concentration gradient. We are creating a massive stockpile. The outer chamber becomes packed with positive protons. They're crowded, they repel each other, they build up a huge electrical and chemical pressure. They desperately want to get back in.
That's the water rising higher and higher behind the dam.
Precisely. You've built up this huge potential energy.
Yeah.
And there's only one way back in for those protons. The membrane is totally impermeable to them except for one very special spot.
The turbine.
A massive, complex protein structure called ATP synthase.
Describe that. What does it look like?
It looks like a mushroom or a doorknob protruding from the membrane into the inner matrix. And it acts like a literal turbine. It physically spins.
It actually spins, like a motor.
It creates mechanical rotation. It's confirmed. As the protons rush back through this channel, driven by that immense pressure gradient, they force the turbine to spin at an incredible speed.
Wow.
And the physical mechanical energy of that rotation is used to smash a molecule of ADP and a loose phosphate group together to create a high-energy bond, forging a new molecule of ATP.
That is mind-blowing.
Yeah.
We are literally using a proton-powered nanoscopic rotational motor to recharge our cellular batteries.
It is some of the most beautiful and intricate machinery in all of nature. It's breathtaking.
So what happens to the electron? It reached the end of the chain. It fell down all the stairs. They can't just hang out there at the bottom.
This is why you breathe. This is the only reason you inhale oxygen every few seconds of your life. The final step.
Okay.
Cytochrome A3, the last protein in the chain, hands those tired, used-up, low-energy electrons to a molecule of oxygen. The oxygen also picks up some of the protons that are flowing back in through the turbine.
So oxygen plus a couple of electrons plus a couple of protons.
Equals H2O. Water.
So the oxygen we breathe in just sits at the very end of the assembly line to take out the trash, the used electrons, and in the process, it turns into metabolic water.
Without oxygen to act as the final electron acceptor, the entire chain backs up. The electrons have nowhere to go, the pumps stop, the proton gradient dies, the turbine stops spinning, no more ATP is made.
And you die.
Within minutes, it all comes down to that continuous flow.
Incredible. So let's look at the numbers now. Let's do the accounting. Section 6, the grand tally.
Let's add it all up per one molecule of glucose.
Okay. Glycolysis out in the cytoplasm gave us a net profit of 2 ATP.
Check.
The citric acid cycle, the Krebs cycle, gave us another 2 ATP directly.
Check, we're at 4.
And this oxidative phosphorylation, the proton turbine, how much does that yield from all those NADH taxis we filled up?
That process yields, on average, 34 ATP.
34. So 2 plus 2 plus 34. The grand total is 38 ATP molecules for every single molecule of glucose we burn completely.
That's the theoretical maximum, 38 ATP, from the paltry 2 we had after glycolysis.
Now, let's go back to efficiency again. We said earlier that 38 ATP represents about 456,000 calories of stored chemical energy. The glucose molecule we started with contains 686,000 calories.
So if you do the division, 456,000 divided by 686,000, you get an efficiency of 66%.
66%?
Yeah.
Is that good? How does that compare to things we build?
It is astounding. A really good internal combustion engine in a car is maybe 20% to 30% efficient. The vast majority of the energy in gasoline is lost as waste heat. The cell is more than twice as efficient as a car engine. It manages to capture 2/3 of the potential energy in its fuel and store it as ATP.
But that still leaves 34% unaccounted for. Where does that other third of the energy go?
The laws of thermodynamics dictate it must be converted into something else. It becomes heat.
Heat.
And this is not waste in a biological sense. This is your body temperature. The reason you are 98.6 degrees Fahrenheit or 37 Celsius is largely due to the constant inefficiency of the system burning glucose in your trillions of cells.
So if we were 100% efficient,
We would be cold-blooded, we'd be the same temperature as the room we're in, we'd be incredibly sluggish every time the sun went down, we would have to bask on warm rocks like lizards just to get our enzymes working properly.
that is a very cool thought. Our waste heat is actually our life support heating system.
Exactly. The inefficiency is a critical feature, not a bug in the system.
Okay, we've built this incredible high-efficiency engine, but you can't run an engine at full throttle all the time. You'd blow it up or you'd burn through all your fuel in an hour. Section 7, controlling the engine. How does the cell know when to speed up or slow down?
It's a beautifully simple supply and demand system regulated by something called allosteric inhibition.
Okay, fancy words. Break that down for me.
Think of one of the key enzymes in glycolysis, near the beginning of the whole process. It's called phosphofructokinase. Let's just call it PFK.
PFK. Got it.
PFK is one of the main managers of the assembly line. It's a major control point.
Okay, PFK is the boss.
If the cell is full of ATP, If the batteries are all fully charged and you don't need any more energy right now, the ATP molecules themselves actually bind to a special spot on the PFK enzyme and turn it off. They inhibit it.
So if you have too much of the final product, the factory automatically stops production at the very beginning.
It's a perfect negative feedback loop. It prevents the cell from burning precious glucose unnecessarily. Also, another molecule, citrate, which is the first product of the Krebs cycle, can also inhibit PFK.
So if the Krebs cycle is backed up and full,
It sends a signal back to the start saying, hey, we're at capacity down here, stop sending raw materials.
and what if we run out of energy? What's the go signal?
If your ATP levels drop because you're exercising, then your ADP and AMP levels rise. And AMP in particular acts as a powerful activator. It binds to PFK and says, open the floodgates, we need power now.
It's a perfect self-regulating feedback loop. No computer needed, just chemistry responding to the concentrations of products and reactants.
It keeps the ATP concentration in the cell surprisingly constant, regardless of whether you are sleeping or sprinting a marathon. The body fights incredibly hard to maintain that currency supply.
Speaking of sprinting, what happens when we push the system too hard? When demand outstrips supply? Section 8, anaerobic glycolysis.
This is the emergency backup generator. Suppose you are lifting a very heavy weight or sprinting the 100-meter dash. Your muscles are screaming for huge amounts of ATP instantly.
But my heart and lungs can't keep up. I can't deliver oxygen that fast.
Exactly. You can't get enough oxygen to the mitochondria fast enough. So what happens to the electron transport chain?
It shuts down. No oxygen to catch the electrons at the end.
The whole thing grinds to a halt. The turbine stops. But remember, glycolysis, it doesn't require oxygen.
Right. It happens out in the cytoplasm. It can keep going.
It can keep splitting glucose into pyruvic acid.
But you said earlier that glycolysis only makes a net of 2 ATP. That's incredibly inefficient.
It's wildly inefficient, but it's fast. And in an emergency, 2 ATP is infinitely better than 0 ATP. But there is a snag. A chemical snag that would stop it.
What's the snag?
Remember the NAD plus taxi?
The one that carries the hydrogen.
Glycolysis needs empty taxes, NAD plus A, to pick up hydrogen as it splits the glucose. Normally, those taxis drop the hydrogen off at the mitochondria and come right back empty for another load, but the mitochondria is closed for business.
So the taxis stay full. The NADH never gets emptied.
All your available NAD plus very quickly turns into NADH. You run out of empty taxis. And without empty taxis, glycolysis stops. The whole system would shut down in seconds.
You're stuck.
You would be,
Right.
unless you open an emergency trapdoor. This is where lactic acid comes in.
Ah, the burn, the thing you feel in your muscles.
This is the one. The cell takes the pyruvic acid that's building up and it reacts it with the full NADH taxi. It essentially dumps the hydrogen from the taxi onto the pyruvic acid, creating lactic acid.
Why on earth would it do that?
Because it empties the taxi. It regenerates NAD plus from NADH. Now that freshly emptied NAD plus taxi can go back to the start of the glycolysis pathway and help split another molecule of glucose.
So the entire point of making lactic acid is just a chemical trick to recycle the carrier molecule so we can keep making those measly 2 ATPs for a little longer.
Exactly. It's a patch. It buys you time. It allows you to function at a very high intensity for minutes without sufficient oxygen instead of just for a few seconds.
And the source calls lactic acid a sinkhole. Why that term?
Because it diffuses out of the muscle cell into the blood very easily. So it doesn't build up to toxic levels inside the cell instantly. It gets washed out. That allows the reaction to keep going longer than it otherwise could.
But eventually you have to stop, and you have to pay the debt.
The oxygen debt. When you stop sprinting and you stand there panting, you are taking in all that extra oxygen to help your liver convert that lactic acid back into pyruvic acid and then burn it properly or even turn it back into glucose.
Fascinating. So that burn you feel is basically the cost of keeping the lights on when the main power plant is down.
That's a very good way to put it.
Now, we've been talking almost exclusively about burning glucose for energy. But there's another pathway mentioned, Section 9, the pentose phosphate pathway. The source says this is responsible for something like 30% of all glucose breakdown in the liver. That's a lot.
This is an often overlooked pathway, but it's critically important. It operates in parallel with and independently of the glycolysis-Krebs cycle pathway.
What's the point of it, if not for energy? It doesn't seem to make much ATP.
You're right, it's not for burning, it's for building.
Explain that.
The normal energy pathway we've been discussing uses the NAD plus taxi to carry hydrogen for burning in the mitochondria to make ATP. The pentose phosphate pathway uses a slightly different but related carrier molecule, NADP plus Latidon. Note the extra P for phosphate.
NAD plus versus NADP plus grow. What's the difference in their jobs?
To put it simply, NAD plus is for catabolism, breaking things down for energy. NADP plus is for anabolism synthesis, building things up. Specifically, it's used for synthesizing fats from carbohydrates.
Oh, so this is the pathway that helps turn that extra slice of cake I ate into body fat.
Essentially, yes. When your cell has plenty of ATP and it doesn't need more energy, the body shunts glucose into this alternate pathway. It breaks the glucose down in a different way to create a supply of NADPH, which provides the high-energy hydrogen needed to build fatty acid chains from scratch.
And that explains why this pathway is super active in the liver and in fat cells.
Correct. That's where most of the fat synthesis happens. This pathway is the construction crew, not the demolition crew.
Okay, so we've talked about burning sugar for energy, and we've talked about turning sugar into fat for storage. What if we go the other way? What if we run out of sugar entirely? Section 10. Gluconeogenesis.
Making new sugar.
Making sugar from scratch.
This is the body's ultimate defense against starvation. Your brain, as we mentioned, needs glucose. It's very picky. If your blood sugar drops too low for too long, you go into a coma and die.
So if I haven't eaten in, say, 24 hours, my liver's glycogen stores are probably gone.
Yeah.
Where do I get glucose from my brain then?
You make it. The liver is an amazing alchemist. It starts grabbing other molecules from around the body. It can take amino acids from your muscles. It can take the glycerol backbone from your fat stores.
It turns protein into sugar. How does that even work?
Yes. For the amino acids, it performs a process called deamination. It strips the nitrogen group off them, and the remaining carbon skeleton can be fed into the metabolic cycle and reversed, essentially, to synthesize brand new glucose molecules.
And there's a key hormone that drives this process, right?
Yeah.
Cortisol.
Yes, the infamous stress hormone. When your body is under prolonged stress, like starvation, the pituitary gland signals the adrenal gland to release glucocorticoids, the main one being cortisol.
And what is cortisol's job in this situation?
Cortisol mobilizes the supplies for this process. It actually signals your muscle cells to start breaking down their own protein and release amino acids into the blood. It tells the liver to ramp up its machinery, grab those amino acids, and start baking bread, start making glucose to keep the brain alive.
That sounds incredibly destructive. You're cannibalizing yourself.
It is catabolic, absolutely. You are breaking down your own valuable muscle tissue to feed your brain. But from an evolutionary standpoint, it's better to lose some muscle mass than to die. It keeps you alive during a famine. It repairs the glucose bank when there are no deposits coming in.
Finally, let's just quickly check the dashboard. Section 11, blood glucose context. What are the normal numbers we're actually trying to maintain here?
In a normal, healthy fasting person, someone who hasn't eaten in a few hours, blood glucose should be about 90 milligrams per deciliter of blood.
And right after I eat that turkey sandwich,
It'll go up maybe to 120, 140 milligrams per deciliter for an hour or so. But it rarely goes much higher in a healthy person because of that powerful insulin response and the liver's ability to soak up the excess.
the liver really is the hero of this whole story, isn't it? It acts as a glucose thermostat.
It absolutely does. It absorbs excess when levels are high, turning it into glycogen. And it releases glucose when levels are low, either from glycogen or by making it from scratch. It works tirelessly to keep that blood glucose line steady for the rest of the body.
So let's bring this all back to the big picture, back to where we started. We started with a turkey sandwich.
We did. A fine sandwich.
That sandwich was chewed, digested, and the carbohydrates were absorbed into the blood as glucose.
Step 1, the delivery.
Insulin opened the door, and the glucose entered the cell.
Step 2, cellular entry.
It was immediately trapped by phosphorylation. Then, depending on our energy needs, it was either stored safely as glycogen, or it was split in half by glycolysis into pyruvate.
Generating a tiny, tiny bit of ATP along the way.
Then that pyruvate entered the mitochondria, went through the Krebs cycle, which wasn't about making ATP, but about stripping out all the high-energy hydrogen electrons.
Loading the taxis.
And finally, those electrons went down the slide of the electron transport chain, powering the proton pumps that spun the ATP synthase turbine to make massive, massive amounts of ATP.
The grand total of 38 ATP per glucose. A big payoff.
And that ATP, that currency, is what allowed my vocal cords to vibrate and say this sentence right now.
It is the spark of life, powering every single thing you do and think.
It really highlights the incredible duality of our system. We are constantly balancing catabolism, the breaking down of fuel, and anabolism, the building up of our bodies.
And it's all driven by that simple ratio of ADP to ATP. The currency supply dictates the entire economy of the cell from moment to moment.
It's truly amazing to think that these complex chemical storms are happening, perfectly regulated, in trillions of our cells simultaneously, just so we can sit here and have this conversation.
And doing it with a staggering 66% efficiency.
Which brings me to a final thought for you to chew on. We talked about how 66% efficiency is amazing compared to a man-made engine. But it still means 34%, a full third of the energy, is lost as heat.
Yes, that's just the price of doing business, thermodynamically speaking.
The source material presents this as just a fact of life. But think about what it implies. Our inherent inefficiency is the only reason we can survive in cold climates. If evolution had somehow managed to create a 100% efficient system for processing glucose,
We would need far less food. We'd be incredibly economical creatures. A single meal might last us for days.
but we'd freeze to death in a slight breeze.
We would. We would be completely dependent on external heat sources. We'd be slaves to the weather, just like reptiles. Our entire civilization, our ability to inhabit the entire globe, is predicated on this flaw.
So in a way, our biological imperfection is actually our superpower. It's our built-in, non-stop personal heater.
That's a beautiful way to put it. The heat of life is just the friction of the machine.
I love that. The heat of life is the friction of the machine.
Ah.
We'll leave you with that thought. Thanks for diving deep with us into the metabolism of carbohydrates.
It's been a pleasure.
See you next time.